You know, for decades, the fight against cancer has been all about reading the enemy's battle plans, our own DNA. But what if we could do more? What if we could actually listen in on their real-time communications? Today, we're digging into a new frontier in cancer therapy that does exactly that by tuning into a molecule called RNA. Okay, so to really get this, let's use an analogy. Think of it like building a house. Your DNA, that's the master blueprint. It's the permanent, static plan for every single cell. But RNA? That's different. That's the set of active work orders that get sent to the construction crew today. It tells you what's actually being built or torn down or modified right this second. Now, in cancer, knowing the blueprint is absolutely critical, but intercepting these moment-to-moment work orders, that could change everything. All right, so let's start with where we've been. For the past decade or so, reading that DNA blueprint has been the absolute cornerstone of precision oncology. It's been a real revolution, for sure, but as we're about to see, it's a revolution with some serious limits. And look, this approach has been a massive success, no question about it. By finding specific flaws in a tumor's DNA blueprint, we've developed an incredible 138 targeted drugs that have been approved by the FDA. These drugs are designed to go after those exact mutations, and for many patients, the results have been amazing. But here's the catch. This just doesn't work for everyone. Sometimes you look at a patient's DNA and you just don't find any of these known targetable mutations. Or maybe you find a mutation, but it's not really clear if it's the one actually driving the cancer. The blueprint, as powerful as it is, doesn't always have the full story. So that's the big question, right? How do we get more detail? And that brings us to this landmark study. Researchers over at MD Anderson decided, what if we just listened to the RNA's message? In a study they called IMPACT2, they analyzed both the DNA blueprint and the RNA work orders, side by side, to see what new secrets would shake out. Okay, let's be super clear about what RNA profiling is all about. While your DNA tells you what a cancer cell could do, RNA profiling shows you what it is doing right now. It's like a dynamic snapshot of which genes are flipped on or off and how loud they're shouting their instructions. It really captures the real-time chaos that drives a tumor. So what they did was pretty comprehensive. They took 253 patients, all with advanced cancers, and for every single one, they performed a really deep analysis of both their tumor's DNA and its RNA. This gave them a direct head-to-head comparison between the blueprint and the actual work orders. And what they found? Well, it's fascinating. The first huge discovery was all about these hidden connections they uncovered between the two layers of information. Now, in some cases, things lined up exactly how you'd think. In 50 patients, a flaw they found in the DNA blueprint of one gene directly matched up with abnormal activity in that same gene's RNA. We call this concordance. The work order perfectly reflects a known flaw in the plan. This confirmed the link, for sure. But honestly, that wasn't the big story here. Now, the real game changer was in the surprises. The analysis found 123 significant connections where a DNA mutation in one gene was linked to weird activity in a completely different gene. I mean, imagine finding out a typo in the blueprint for the electrical system is somehow causing the plumbing to go totally haywire. This revealed a whole hidden network of cause and effect that a DNA-only test would just completely miss. Let me give you a really powerful example of this. A mutation in TP53, which is one of the most famous or maybe infamous genes in cancer research, was found to be strongly connected to the hyperactivity of a totally different gene called VEGFA. So why does this matter? Well, VEGFA is the gene that helps tumors build new blood vessels to feed themselves. So this finding suddenly creates a new actionable link. If you see a TP53 mutation, maybe the real way to attack it is to target the VEGFA pathway. Okay, but that wasn't all. The second huge finding from this IMPACT2 study gave us a brand new tool, a way to actually measure the overall chaos level inside a tumor just by looking at its RNA. And they gave it a name, Tumor Transcriptional Burden, or TTB for short. The best way to think about it is like the amount of molecular radiostatic, or just pure chaos, inside the tumor. It's a really simple count. How many genes have abnormal RNA levels? So a low TTB is like a clear radio signal. Things are relatively orderly. But a high TTB, that's just a wall of static, a storm of confusing, contradictory work orders being shouted all at once. So why does this molecular chaos matter so much? Well, the researchers plotted patient survival against their TTB score, and the results are just stark. I mean, just look at this chart. That green line, that represents patients with the highest level of molecular chaos, 6 or more genes with messed up RNA. And their survival curve just drops off a cliff compared to the others. And the numbers behind that line, they're brutal. For those patients with the highest TTB score, the median survival was only 6.7 months. This shows that TTB isn't just some random measurement. It's a really powerful new prognostic tool that can help predict a patient's outcome based on the real-time biology of their cancer. You know, the researchers themselves put it perfectly. They concluded that RNA profiling isn't just some interesting academic thing. It provides concrete, actionable information for doctors and patients that goes way beyond what they could ever learn from just looking at DNA alone. Okay, so let's pull all this together. What does this actually mean for the future of cancer treatment? Well, it means we're moving toward a much richer, more complete, and ultimately a much more actionable picture of the enemy we're fighting. All right, so let's boil this all down. What are the big takeaways here? First, we have to remember, DNA is the blueprint, but RNA is the real-time action plan. Second, by listening to that action plan, we can find these hidden gene connections that DNA-only tests just can't see. Third, we have this powerful new tool, TTB, to actually measure a tumor's molecular chaos and get a sense of prognosis. And finally, when you put it all together, combining both the blueprint and the action plan gives doctors a way more complete picture of an individual's cancer. You know, all of this research really leads us to one massive question for the future of medicine. We've gotten really good at reading the blueprint, but now that we're learning how to intercept the real-time work orders, will this two-pronged approach become our new gold standard in the fight against cancer? Based on this evidence, the answer is pointing in a very clear direction.